Morning. This lecture video is going to go over impulse transmission. So this is going to be what has to happen chemically and electrically to the neuron to get an impulse to go from the dendrites all the way down to the axon terminals. And then we'll talk about what happens down in these axon terminals to go from this neuron to the dendrites of the next neuron. So you can follow along on the diagram. This is kind of the very easy, abridged version of what's happening. So, keep in the back of your mind that this is a little bit more complicated than what I'm making it, but I'm trying to make this as simple as possible. So just to orientate yourself, this entire structure is the neuron. So this is going to be the dendrite, right, bringing the signal in. This is going to be the cell body and the nucleus. And then this is going to be the axon. So kind of write that here. These are going to be the axon terminals. Now, to kind of just go over these structures here in the axon terminals, I'm going to actually put this right here. This is called a vesicle or a synaptic vesicle. And inside the synaptic vesicle is going to be chemicals called neurotransmitters. Your neuron, remember, is a cell. So all of this in black is a cell membrane, just like the cell membranes we talked about when we went over the parts of the cell. An impulse transmission is going to occur because of different electrical and chemical changes along or across that cell membrane. And there's basically like 4 different steps here. So I'm kind of going to divide my neuron into these sections in order to kind of help organize what's going to be happening here. So we're going to start with step number 1, which is called the resting potential. The resting potential is going to be the normal environment, the normal chemical and electrical environment of the neuron. So this is going to be when it is not conducting an impulse. In this normal environment, we are going to focus on 2 ions. We're going to focus on sodium, and we're also going to focus on potassium. Now, sodium and potassium are both positively charged, so that means that they gave up an electron. However, they're not evenly charged, like the charge isn't the same. So actually, your potassium is more negative than the sodium. So what's going to happen is that your sodium, there's going to be sodium that's going to be concentrated outside of the cell. And the potassiums are going to be concentrated inside of the cell. So if I were to draw these up here, here we go. We got a bunch of potassiums inside, right? There are some floating around on the outside too, but there's more of them on the inside. And if I draw the sodiums, we have more sodiums on the outside than we do on the insides. What happens then is that this is going to cause a difference in charge from the outside to the inside. So the inside of the cell at resting potential is more negative than the outside of the cell, which is more positive. Okay? So if I continue down, inside is negative, outside is positive. And we can actually measure this. It's negative 70 millivolts. More negative on the inside than it is on the outside. So this is what's going to be the normal environment of the cell when it's not conducting an impulse. The next thing that happens is, and we'll put this over here, is going to be the dendrites are going to receive a stimulus. And it has to decide whether or not it's strong enough to continue the stimulus and to continue the impulse down the axon. And this is called a threshold. Your body, your neurons, receive impulses and stimulation all of the time. But oftentimes it's not strong enough to continue the impulse down the axon. And that's a good thing, because if we sent impulses down the axon for every time there was stimulation, we would literally go crazy because our brain would be constantly sending these signals, and we don't need to. So if the signal, and this is going to be like it's going to be negative 55 millivolts if we measure what the threshold is in a normal neuron. If that signal is stronger than that, then it's going to trigger a series of events which we call an action potential. And that basically an action potential is the word that we use to describe impulse transmission. So this whole process going from resting potential to depolarization to repolarization to refractory, this is all going to be considered an action potential. So we're going to assume then that the stimulus is strong enough, it's reached that threshold. So what's going to happen is the next step, which is called depolarization. During depolarization, the sodium channels or sodium gates are going to open, and sodium is going to rush into the cell. And eventually, the inside is going to become more positive than the outside. So we'll draw our channels in here, right? We're going to draw, again, our sodiums on the outside. That's where they started. And we had sodiums on the inside. So now these sodiums are going to rush into these gates because these gates open up. We still have our potassium, that's all on the inside. And we're going to end up now with a whole bunch of sodiums on the inside. And this is going to make the inside of the cell positive and the outside of the cell negative. Okay? So it's like opening the door to a sodium party, right? They're all rushing in. Awesome. Now, just like any other party, sometimes you overstay your welcome. So what happens in this next stage, which is called repolarization. So notice now during depolarization, we've changed the chemical environment, right? Because now that we have a different chemical makeup of the inside of the cell, but we've also changed the electrical environment. So we often call this an electrochemical gradient because we're changing not only the positive and negatives, but we're also changing the, the chemical composition because now we have sodiums on the inside and we have less of them on the outside. Once this happens then, the next stage is called repolarization. And what happens during repolarizations is that the sodium gates are going to close and potassium gates are going to open. And potassiums are going to rush out. So again, we've got all of these sodiums floating on the inside. Sodiums, okay? We've got just a couple left on the outside. We have all of these potassiums on the inside, and these are going to now start to flow out to the cell, outside of the cell. So we're going to end up with all kinds of potassiums on the outside of the cell. And this again is going to cause a swap and change. So now we're going to end up being more positive, and the inside's going to end up being more negative again. You can tell this is going to be triggered, so this will actually happen, and when this happens, it's going to cause the insides to be plus, let's just say 40 millivolts. And then once it reaches this point, that's what's going to trigger these gates to open, and this will end up driving the reaction, is this change in charge. So now we have our charge distribution that's back to normal, right? Because this is our resting potential. So it's positive on the outside, but it's negative on the inside. But we have not put the chemicals back in order. And so this last stage is called the refractory period. So during the refractory period, what's going to happen is that a pump that's called the sodium-potassium pump is going to pump the sodiums and the potassiums back to resting potential position. And this requires energy. So we're basically going to go back to this. Your brain, maintaining this resting potential takes so much energy that it's estimated that 40% of the energy that your brain uses is using to maintain the sodium-potassium pump. The neuron cannot accept another impulse until it has gotten back to resting potential. So impulses are all or nothing, because once this starts, once we hit our threshold, depolarization, repolarization, and refractory are going to occur regardless. It's all or nothing. It won't happen at a weaker or stronger. It's just going to go through. But in order for it to receive another signal, we have to get it back to resting potential. So how quickly we can send that signal has to do with how quickly that sodium and potassium pump can bring it back to the resting potential, so that once it's at resting potential, it can again accept a new stimulus and pass that along. So this is how the impulse travels from the dendrites all the way down the neuron down to the axon terminals. Resting potential, depolarization, repolarization, and then refractory period. Charges switch, chemical balance of the ions switch. And then eventually they're going to go back to where we want it to at resting potential. So now what we do is we need to talk about what happens when we get to the end here to take that impulse and move it to another neuron, to the dendrites of another neuron. So if you flip this sheet over, this is going to kind of explain this process. So again, here is our vesicle, and inside here is our neurotransmitters. So if you want to label those to help you remember. So here's the vesicle, the synaptic vesicle. Synaptic. And it contains neurotransmitters. There are several different types of neurotransmitters, so not all neurotransmitters do the same thing, but you can kind of get the general idea. Now, we have talked about the importance of sodium, we've talked about the importance of potassium, but there's another chemical that's floating along on the outside of the cell that's going to play a role in transferring the signal. So here's the axon terminal of one neuron, and this is showing you a blown up version of the dendrites of another neuron. And so here are going to be, we're also going to just call these gates or chemical gates. In between is called the synaptic gap or the synaptic cleft. It's called both. And so it's going to be the area between two neurons. So here's what happens. The impulse comes in. So here's the impulse. So again, depolarization, repolarization is occurring. And what's going to happen is step one, when we get down to the bottom, is going to be that these calcium gates are going to open. So there's more gates that open for calcium. So they are different than the potassium and the sodium gates. And calcium is going to rush into the cell. So calcium's going to rush in, right? So we got all these calcium now floating around inside the axon terminal. The calcium is basically going to tell the vesicle to move to the end of the cell membrane and merge. So this vesicle is going to end up coming over here, and it's going to end up kind of merging with the cell membrane, and it's going to dump all of these neurotransmitters now into the synaptic cleft. So 3 is the neurotransmitters are dumped into the gap. Those neurotransmitters then are then going to bind to gates on the dendrites. Once they bind, one of two things will happen. So the first thing that could happen is that it's going to trigger sodium gates to open, which, let's go back, right? We triggered sodium gates to open here, so now we're going to depolarize, right? So it triggers sodium gates to open, and depolarization occurs. And we get impulse transmission, right? The impulse will continue down the next neurotransmitter. The other thing that could happen, and this is going to depend on what kind of neurotransmitters there are. Some neurotransmitters are stimulatory. So this is what's going to happen here. So we'll just put stimulatory on here. Some neurotransmitters are inhibitory, meaning that they're going to stop the flow of dendrites. Stop the flow of the impulse. So what happens with these is that you're going to end up triggering potassium gates to open. These are called inhibitory neurotransmitters, and I'll tell you why here. So I'm going to flip this over again quick. If this triggers potassium gates to open right here, what happens is that it's going to take a lot more time for the sodium and potassium pump to get it back to resting potential, which means that the transmission of the impulse is going to be slower or stopped. because we've opened up these gates now, so it's going to take, it's like, you know, if you open up a gate to 1,000 animals and they all rush out, it takes longer to bring them back into the pen. It's the same general idea. If we open this potassium channels, it's going to take so much longer to get back to resting potential that the impulse is either stopped in its track or it slows down a ton. So these are called inhibitory neurotransmitters because they're going to slow down the transmission of impulses, versus a stimulatory one, which is basically going to trigger depolarization. And so this is how impulses go from one neuron to another. It's a chemical bridge, and that chemical bridge is accomplished through these chemicals called neurotransmitters. Super easy version of impulse transmission. It's a little bit more complicated than this, but these are kind of the basic steps that are involved that we want you to know. I know your brain is probably mush right now, and I apologize for that, but if you have questions about this process, make sure to ask. It's a lot easier to go through it this way than trying to understand it through the textbook, just because your textbook is going to get into a lot more of the nitty-gritty details. And what I just really want you to know is this basic process of, you know, resting potential, depolarization, repolarization, that refractory period, and then how the impulse is going from one neuron to the next neuron. The role of sodium, the role of potassium, the role of calcium. So these are important. They all play a really important role. And then also the chemical role of the neurotransmitters. And then also knowing how that change in electricity or electrical charge shifts during this process along the cell membrane. Have a great day.